Well, welcome to the fourth session of today's symposium. Our first speaker is Professor William Jungers, organizer of this meeting. Bill's a long-time colleague and friend. He's also a distinguished teaching professor. It's getting to be a long day. Distinguished teaching professor and chairman of the Department of Anatomical Sciences in the medical school here at Stony Brook. A native of Texas, Bill received his PhD from the University of Michigan in 1976 and has been at Stony Brook since 1978, a lot longer than he'd probably care to admit, but I'll admit it for him. Bill is best known for his work relating to body size and allometric scaling in primate and human evolution, certainly issues of relevance given today's topic and the nature of the beast under consideration. He has a particular, some might even say peculiar, interest in very large subfossil lemurs from the island of Madagascar and very small subfossil hominins from the island of Flores. I can't figure that one out. He is deeply involved in the description and analysis of the skeleton of Homo floresiensis, and most significantly for us, Bill is responsible for organizing today's symposium and arranging for the first public display of the Hobbit outside of Indonesia. We're all very grateful to you for this, Bill. Dr. Jungers, can island dwarfing explain Hobbit body size and shape? Bill.
Thanks for sticking it out, gang. I appreciate it. As many of you probably know, certainly anyone who's lived on Long Island for a while, strange things can happen on islands. You can sort of see the cosmic intersection of Long Island and Flores here. As Mike Morwood mentioned this morning, sometimes small animals get big on islands, and sometimes large animals get small on islands. This has been codified into an evolutionary rule by some people. It's the island rule, if you would. And really what I want to revisit today is the question, can the island dwarfing, or the island rule, explain Hobbit body size and shape, or Hobbit body size and design? I think the answer to the question, whether it's yes or no, is going to be interesting, and it'll be pretty clear once I get started, sort of where I'm going to place my bets on the answer to this. In the initial and amazing announcement of this new species in 2004 by Brown, Morwood, Sutikna, and colleagues. Obviously, one of the things that caught everyone's attention was the size of Homo floresiensis, and in particular what we knew about LB1, the type specimen. What the authors suggested in that paper was that the most likely explanation for its, that is Homo floresiensis, existence on Flores is long-term isolation, with subsequent endemic dwarfing of an ancestral Homo erectus population, presumably Homo erectus from Southeast Asia. And so in a sense, in this first paper, they were suggesting that here was a bipedal hominin that was following something now known as Foster's Island Rule, after a very famous paper by J.B. Foster in Nature in 1964. I think it's fair to say that if you were to survey many professionals who actually recognize Homo floresiensis as a new species, it's this point of view, this explanation would resonate most positively. I think many people would, if they had to vote, that's probably where most people would vote. In 2008, there were a couple of more papers that were published that tended to support this idea. Van Heteren published a paper called Homo floresiensis as an island dwarf, wherein he argued that Homo floresiensis was a pedomorph, that is, a dwarf that had sort of juvenile characteristics, that its growth had been truncated to account for its small body size and peculiar morphology. Also this year, in 2008, Lyris et al. Sort of also agreed with this, that this is the most likely explanation for certain features like a relatively large tooth size and some peculiar aspects of body shape. So again, these authors were suggesting that, like Stegodon on Flores, the hobbits were a dwarf descendant of Homo erectus, supporting the original conclusions by Peter Brown and colleagues. Now, there's a few ironies to this. One of the ironies is that in the same year, 2008, it's turned out that the island rule is pretty unruly. In fact, some people have gone so far to suggest that it may be an artifact, that in fact there are only a few species, carnivores, some rodents, and artiodactyls, that sort of predictably dwarf on islands. And the other cases of dwarfing can't really be predicted by group. So again, I think this hasn't really penetrated the literature yet, but I think it has some profound implications for sort of the assumption that if it's small, it must be a dwarf. A greater irony, however, comes from the second paper that Morwood, Peter Brown, Sutikna et al. Published the year after, when they returned and found new skeletal remains of Homo floresiensis, continued their analyses, and they began to change their opinion, and I think changed it in a fairly profound way, when they concluded that the genealogy of Homo floresiensis remains uncertain. Similarities in stature and body proportions with Australopithecus, for example, may reflect phylogeny or secondary evolutionary convergence. Either way, Homo floresiensis is not just an allometrically scaled down version of Homo erectus. For some reason, this seems to have received or gained less traction in the literature. I don't know, I don't think people ignored it, but I think people were more comfortable with the island dwarfing notion because this solution to the question, this answer to the question I posed in the beginning, has very profound biogeographical implications, and I think some people are still reluctant to embrace those, and I'll talk a little bit more about those momentarily. Just as context, there are, as you've already seen today, I hope, there are basically two fundamental differences in body design in human evolution. Sort of the primitive body design is a short and stocky hominin, bipedal to be sure, small brain, wide flaring hips, and relatively short hind limbs. The derived condition, the presumptive condition from which the hobbits were derived, if dwarfed at all, is seen here in the early Homo erectus from Africa, sometimes called Homo ergaster by some folks. Here we have a long, linear body design, and in particular, we have relatively narrow hips and relatively and absolutely very long hind limbs. Some major differences in what we see from this condition. In many respects, but obviously not all, as Susan Larson talked about earlier, this design is very similar to our own in many respects. Sort of the wild card in all of this is Homo habilis. You can see there's a lot of pieces to the puzzle, but it's rather hard to put together. But there is some emerging evidence recently to suggest that Homo habilis may actually share many aspects of body design with Australopithecus as opposed to Homo erectus. Let me put a little finer point on this. And that is to say, we now have two pretty good skeletons of Homo erectus. We have the Nereocotomy boy in East Africa. We have it even at an earlier time, the Homo erectus from Dmanisi. You can sort of see their locations here. The question is, do the Hobbits show any signature, any skeletal signature of Homo erectus? And if they don't, what possible evolutionary transformation correlated with dwarfing could account for all of the evolutionary reversals to take something like this and make it like this, which in many respects is like Australopithecus, if not Homo habilis. Now, I have nothing against ungulates. I just want to get that straight now. But I don't think they necessarily make the best model for predicting what a dwarfing, relatively large-bodied bipedal hominid is going to look like. Wouldn't it be wonderful if we had bipedal hominins that had dwarfed from a larger body size to sort of give us a hint some suggestions of what degree of morphological change tends to be correlated with decreases in body size. It turns out we have lots of examples of that. They're called human pygmies. Pygmy is not a pejorative term. It just means short people. And short people have evolved independently, their body size and design, throughout the world. In South America, in Africa, in Asia, sometimes on islands, sometimes on the mainland. So the question we can ask today is, do pygmies inform us in any meaningful way as to what we might expect, what tendencies we might anticipate going from large body size in a modern design to just smaller body size? And I don't want to tip my hand too much here, but I can tell you right now, it's going to be obvious that pygmies are still very similar to you and me. The differences are subtle in their proportions, and I think that that has profound implications for how we interpret the ultimate morphology we observe in Homo floresiensis. So just how big is Homo floresiensis? I'm going to focus primarily on LB1, the type specimen, because we have the best sources of skeletal remains to use as estimators. I estimate body mass based on joint size, hind limb joint size, things that support the body weight of an individual, as 30 to 35 kilos. And for those metrically challenged individuals in the audience, that's about 66 to 77 pounds. This is only slightly larger than what Brown and Morwood and colleagues had published in the beginning, but just a little bit heavier. Actually, there are smaller individuals. LB1 turns out to be kind of the Hulk of the Hobbit population, if you can believe that. Most of the other individuals are slightly smaller, but again, sort of I would say in the 26 to 30 kilogram range. In terms of body height or stature, however, Homo floresiensis is genuinely short. I estimate it to be just over a meter, maybe 1 meter 0.06. Again, translating to about 3'4" to 3'6", and this depends a little bit on which statistical model you used and which reference population. But I think it's fair to say that Homo floresiensis was indeed short, as we can see in all of these sort of illustrations in front of you. Now, let's put this in context. Let's sort of throw some data at the situation and ask the question. What I've put together for you is a frequency distribution of body mass, and this is just in female pygmies from Africa and from Asia, and some fossil hominids. Did I back up one? No, I guess that's okay. And what we see right now is that here's the frequency distribution of body masses. The range is from just around 27 kilos to about 50 kilos. And here's LB1. No problem. You can find humans of LB1's body mass, at least, without any problem. What's interesting is the small Homo erectus from Dmanisi, presumptive female, by these criteria would also be characterized as a pygmy. Now that means if we derive Homo floresiensis from a small Homo erectus, there's only about a 20% body size reduction to be required at all. Not very much. So sort of the starting point is critical here. And that's why I want to put these earlier hominids to the right for you to take a peek at. McHenry's estimates for female body size in Australopithecus and in Homo habilis are spot on for what you see in Homo floresiensis. This means if these were the ancestral stock, or the ancestral stock from which we derive Homo floresiensis, then there's no body mass reduction required whatsoever. So sort of keep that in mind. However, in terms of stature, let's look at that same sample of 147 individual humans who are classified as pygmies. Here's their frequency distribution over here. Once again, the small Dmanisi Homo erectus qualifies as a pygmy, about 4'7", 4'8". Here's LB1, and just for grins, Lucy, AL28-1. So LB1 and Lucy are much shorter even than the shortest human pygmies. Now let's do something simple. Let's combine that information about body mass and stature into a simple measure of body shape. That is, how much mass is there sort of per unit body height. And that's just a simple index called the ponderal index. It's like how you pack your weight onto your skeleton. It's just the cube root of body mass divided by stature. And what I've plotted for you here is a large group of human mean values from this source, no pygmies included here to start with. And this is sort of the range of humans. Humans tend to be relatively tall and narrow, relatively lean, as exemplified by this female. Whereas individual apes, there's not a lot of data out there, but individual apes tend to be outside of the range of humans. Apes are packing on much more mass per unit stature than humans. Apes are stocky compared to humans. And I'm afraid to plot myself in this, but I'm hoping that I'm somewhere in this distribution. Now, let's add a fossil or two to this story. And again, we can add the two Homo erectus, strapping youth, and the small Dmanisi fit right in this distribution. And notice I've added to those human means 347 individual pygmy values, which completely subsume the other groups. Human pygmies have the same body shape as other people of taller stature. Here's Lucy. There's LB1. They are both plotting with the apes. They are stocky. They have a lot of mass for their estimated stature. So all the Homo erectus falls within modern human standards. It's clear that these fossils do not. And human pygmies, more importantly, do not become more ape-like as they reduce in body size. So that would suggest if we're evolving from Homo erectus, let's say if Lucy were an ancestral stock, we'd have to go in one direction and it would require an evolutionary reversal to return to that body shape in Homofloresiensis. Let's look at another aspect of body shape, just a simple. Let's just look at sort of limb proportions, the relative lengths of the upper limb and the hind limb. Along this axis, it's just sort of the ratio of your arm to your thigh. And on this axis, it's going to be the ratio of your forearm to your leg. And what you can see plotted here in black dots and a black ellipse are humans of sort of normal stature. And in red, this is a large collection of human pygmies from all over the world. And I think you can see that human pygmies do not converge on the limb proportions of Homofloresiensis. Homofloresiensis has very short hind limbs, and that's what's driving this relationship. It's what's driving the stature I talked about as well. And Peter Brown demonstrated this very nicely in diagrammatic form just a year ago. Short hind limbs is the story for LB1, and pygmies do not appear to move in that direction. So let's put the fossils back on again. Now, we don't have many forearms to plot, but we can plot this ratio, humeral-femoral index, along here. Again, arm to thigh ratio. Same plots as before with human pygmies and other humans. Here's the Dmanisi Homo erectus. There's the Homo erectus out of neurocotomy. Here is Lucy, and there's LB1 again. So once again, there is no hint that pygmies move in the direction of this stockier ape-like body build, whereas LB1 is reconverging on Australopithecus afarensis if they are coming from a Homo erectus-like body design. So again, we have to invoke not just body size reduction, but evolutionary reversals to account for the morphology, the shape of Homofloresiensis. Now, this is one of my favorite aspects of the body design of Homofloresiensis. This is the reassembled foot of LB1. We don't have the heel bone, but it's pretty easy to estimate the total length of the foot. And here is the tibia, here's the femur, and you might notice that foot looks a little big for those elements. But What's driving this again is a very short hind limb. So let's look at the ratio of foot length to femur length, just as an aspect of shape we can capture relatively easily. Let's get relatively tall people. Let's add a collection of human pygmies to that. And what you see is the percentage of foot length, fleshy foot length, relative to thigh length, is pretty much the same. There's no change. However, there's Homofloresiensis. And what does that look like? It looks like the pygmy chimpanzee, Panpaniscus. So this is yet another case where we know that Australopithecus afarensis also had relatively long feet. We're not sure how long they were, but this would again suggest a evolutionary reversal of some magnitude to go from a modern human-like foot, at least, to a LB1 foot. And we'll talk about Homo erectus in just a second. There's other things about this foot that are important for you to notice. It probably didn't have an arch. We think this part of the medial side of the foot was probably weight-bearing. And the intrinsic proportions, the shape of the foot, is also very, very unusual. So without going into too much detail, I want to hear, these are the punchlines. And for the data-oriented folks in the audience, these are chimpanzees. These are humans, including very small body size humans. The relative size of the big toe falls within the chimpanzee range, not in the modern human range. That's a bit of a surprise. And the toe bones themselves tend to be relatively long and curved in Homofloresiensis compared to modern human feet. Here's the human relationship. Here is the chimpanzee condition. This is a very unusual foot for a bipedal hominin. And in fact, I think it has certain implications for how hobbits moved. Let's see if we can go back to that. Is there any way to start this, is the question. Can you give me a hand here? Can you start my, I can't seem to start the video. Yeah, can you do it? Bingo. There we go. Thank you very much. All right. What does it look like? Now, this is, sort of still in progress, but that long foot does something quite unusual. It results in a fairly high-stepping gait and a kinematic profile that's very different than yours and mine. So I think it's fair to say that hobbits walked a little differently than you and me, and they have a foot design that clearly is not made for running. This is not your endurance runner that Lieberman and Bramble like to talk about. Now, I wish I could tell you we had tons of Homo erectus feet to compare, but it's like Matt Tocheri talked about earlier with wrists. We don't have much to go on. But the Dmanisi feet, there are a few bones, and the authors of those descriptions have suggested that it's very modern in aspect, that had an arched foot, etc. And so even though we don't have many feet of Homo erectus, we now have footprints of Homo erectus. And the authors of this study conclude that you have an essentially modern human foot anatomy and function. I don't think that's what we see in hobbits. I think if a hobbit were to make a footprint, it's more likely to look like this one over here. And those are the Latoli footprints, normally attributed to Australopithecus afarensis. Again, if we're going to go from Homo erectus to hobbits, we have to undergo evolutionary reversals in the way their bones are built. Homo erectus has really, really thick bones, especially in the hind limb. These are hobbit bones. They're just like Lucy. They're just like yours and mine. They're relatively thin, normal in distribution. They lack the thickness, which is characteristic of all Homo erectus. Yet another evolutionary reversal. Some features we thought were going to be instructive have turned out to be less so in answering this question. As Susan Larson talked to you today, it turns out that this sort of lack of humeral torsion and short clavicles turns out to be primitive for early Homo in general, maybe early hominids. So that doesn't allow us to choose. There's no evolutionary reversal implied in this particular region. What about in the pelvis? Here's LB1 articulated with Lucy sacrum. Nice fit, reflected. That's what it looks like. I suggest to you that that pelvis, as Peter Brown and colleagues suggested in the beginning, looks like Australopithecus. Here's a modern human. Now, if this is indeed Homo erectus, this is a new pelvis from Gona that is suggested to belong to Homo erectus, although it's not associated with any craniodental material, then like the shoulder, what we're seeing in Homofloresiensis isn't special. And like Australopithecus, it's just preserving the primitive condition. However, if this is Paranthropus, then again, we have another evolutionary reversal. So I've stacked the deck here, but I want you to know there are features that fit the island dwarfing model. And Karen Babb and her colleague McNulty have shown that in craniofacial form, if you do 3D geometric morphometrics and plot size and shape against other aspects of shape, you get humans, you get apes, you get Australopithecines, and you get early Homo. And LB1 is close to what one would predict for a very small or dwarf skull. So there is some evidence, at least in the skull, for the dwarfing scenario. And this has been reproduced by another author, another group of authors, using different methods. Here's LB1, and they also conclude that it's consistent with either Homo habilis or erectus. Here's LB1 linked to Dmanisi, to African Homo erectus and African Homo habilis, distantly connected to Southeast Asian Homo erectus, and that's an important take-home point. This skull could be like a small Homo erectus. So let's just consider the alternatives. I'll close here. If the hobbits are insular dwarves derived from Southeast Asian Homo erectus, then evolutionary reversals need to be invoked from head to toe. We need in brains, perhaps in jaws as well, inside and out, as part of a unique and unprecedented process that would be completely unpredicted by island dwarfing in hominids. And also, there's no special affinities with Asian Homo erectus, regardless. So is it falsify the island dwarfing? I don't know if it falsifies it, but I think it makes it increasingly unlikely. Instead, if Homofloresiensis is descended from a small-bodied, small-brained hominid more primitive than Homo erectus, then an earlier out-of-Africa event that Mike Morwood mentioned is conceivable. But obviously, this needs paleontological confirmation. But this is where I'm placing my bets for now, based on the evidence in hand. And I know where to look. I'd love to see 2.5 million year old fossil deposits anywhere in Asia. But the Suwa Basin that Mike Morwood talked about this morning is going to be critically important in this respect. We know they're fossil hominins on Flores 800 to 900,000 years ago, making tools. All we have to do is find what their bones looked like. If they turn out to be Homo erectus, then I'm wrong. It's happened before. I can live with it. But if it turns out to be something more primitive than Homo erectus, I think this will be great support for an earlier out-of-Africa scenario. Thank you very much for your attention.
Thank you, Bill. I didn't know that you'd ever been wrong. Our next speaker and our last speaker in today's symposium is Professor Charles Hildebolt. Charles is professor of radiology at Washington University School of Medicine and an adjunct professor of anthropology at Washington University in St. Louis. He received his DDS from Ohio State University in 1970, and after 9 years of private dental practice, moved on to obtain a PhD from Washington University in 1987. Charles' research involves the investigation of new methods of digital radiographic imaging that will enable him to obtain measurements of bone mass. These data can be usefully applied not only in dentistry, but also in osteoporosis risk assessment. Charles has used computed tomography, CT scanning, extensively in his research in human health, resulting in a publication record that exceeds 155 peer-reviewed articles. Where do you find the time? Most recently, he has applied his imaging skills to the study of the brain, or more precisely, the endocranial cavity of the skull of LB1 of Homo floresiensis. Charles will speak to us this afternoon on virtual hobbits and health in Homo floresiensis.
Okay, great. Well, as many of the presenters have suggested today, ever since Homo floresiensis was named as a new species, there have been a number of skeptics who have suggested that the skeletal remains of Homo floresiensis are nothing more than the remains of a modern human who suffered from some sort of pathology. And a number of pathologies have been suggested. One group of skeptics think that Homo floresiensis suffered from true microcephaly vera. Now, microcephaly is a complex condition. It involves at least 5 genes and 7 gene loci. One particular type of microcephaly is microcephalic osteoplastic primordial dwarfism type 2, known by the acronym MOPD2, and I'll be referring to that condition several times today. 2 other conditions are Larson syndrome and Cretinism. The skull of LB1 was taken to Jakarta, Indonesia, where it was CT scanned in a medical CT scanner. The details of this aren't too important. CT stands for computed tomography. It's also known as a CAT scan, computed axial tomography. What a CT scan results in is a volume of data or a volume of numbers, and from those numbers, one can create virtual images of whatever has been scanned. In this case, is a virtual skull. And from the inside of the virtual skull, a virtual brain can be constructed, or an endocast. And this technology is a multi-billion-dollar technology. It's used thousands of times around the world every day for making diagnoses, for planning surgeries, for evaluating surgeries. So there's been a great deal of effort devoted to making measurements from CT scans as accurate as possible. And so we use CT scan data to evaluate the brain of Homo floresiensis and also to assess the different pathologies that have been suggested for Homo floresiensis. Now, I'm one of the collaborators on a number of studies that have been performed with the CT scan data. I will give the references. I do want to mention Dean Falk specifically. I've collaborated with her on various brain evolution projects for over 20 years now. And Kirk Smith, who's a senior research engineer at our institute, who created all of the images that I will show today and several other, or Dean showed earlier. And I'll also give the references for the skeptics. You can read the names yourselves. Dean showed this slide earlier. This is a top view of the endocaster brain of LB1. To the left is microcephalic, to the right a Homo erectus, on top a normal human, at the bottom a chimp brain. And these are the side views of the same brains. As Dean pointed out, the brain of Homo floresiensis was least like that of a microcephalic. Microcephalics have pointed frontal lobes. The frontal lobe of LB1 is rather wide. Also, the cerebellum on microcephalic sticks out behind the brain. The cerebellum is tucked in underneath the brain on LB1. The brain of LB1 most resembles that of Homo erectus. They both are wide in the posterior part of the brain, and they both, when viewed in a lateral view, have this long, low lateral profile. So our conclusion was that LB1 didn't have microcephaly. Microcephaly usually results in mentally retarded individuals, and 78% of females are dead by the age of 30. LB1. Not now. Sorry. We now know that there were multiple individuals represented by Homofloresiensis that existed over a time span of thousands of years and hundreds of generations. As I say, our conclusion was that LB1 did not have microcephaly. This has just been discussed. This slide, I think we've seen at least three times today. We did scan in our initial article a pygmy, as was evident in Bill's slides. Pygmies, if you look at pygmies compared with normal humans, they have large heads, and that was the case of our pygmy. It had a cranial capacity of 1,250 cc's. The pygmy is on the left. The brain of LB1 is on the right. Normal humans have a cranial capacity of about 1,350 cc's. Pygmies have brain capacities that are in the range of normal humans. So our conclusion was that we didn't think we could rule out endemic island dwarfing, but we thought that would have taken unusual scaling to have occurred. And we concluded that the ancestor may have been an unknown small-bodied and small-brained hominin, and that had been previously suggested in the original publication that described LB1. Another group, or a group of skeptics, suggested that LB1 was a Rampasasa pygmy who had microcephaly. Now, these pygmies live in a village very close to Leongbua Cave, where the Hobbit was found. And as evidence for this, they said that these pygmies lack chins. I'll show why that's important, and it's been discussed earlier today. And LB1 had severe facial asymmetry, which they said supported that it had pathology. This is a microcephalic skull. Microcephalic humans, they have chins. LB1, as we've seen several times today, Peter Brown's slides specifically, doesn't have a chin. In this article, this soft tissue lateral profile was used to demonstrate that these pygmies don't have chins. It is very hard to assess a bony landmark or bony tissue from soft tissue pictures. Now, if this individual had a chin, then it's highly likely that there would be a bony chin. But because this person doesn't apparently have a chin based on a soft tissue image, is not very strong evidence for this individual not having a chin. The best way to do this would be to use lateral cephalometric or cephalograms, but they didn't present that sort of data. What they did, they made a number of two-dimensional measurements, and one piece of evidence that they used was that they took a two-dimensional image and they mirror imaged the right side of the of LB1's skull and created this image, and mirror imaged the left side of the skull and created this image. And they said, gee, look how dissimilar these two mirror images look. This supports that LB1 had severe facial asymmetry and was pathological. The problem with this is that it is exceedingly difficult to do mirror imaging, particularly with 2D images. It depends on the orientation of the camera and the skull at the time the picture is taken. We repeated this exercise with three-dimensional volume data. And when we did that, there was not nearly the asymmetry that was seemed to be present in the 2D images. Now, this is tricky to do even with three-dimensional imaging. It's done in planning craniofacial surgery, but we concluded that there wasn't a pathological degree of asymmetry. There is asymmetry in LB1 skull, but not a pathological degree of asymmetry. In a recent publication, a very detailed statistical study that Bill just referred to concluded that the asymmetry in LB1 skull is within the limits of asymmetries in normal human skulls and ape skulls. Another group of skeptics published two articles. The second article was an enlargement on the first article. The same points were made in both of the two articles. The major point in their article was that they had a microcephalic skull, or an endocast, from this microcephalic from the Lesotho region of South Africa. The person belonged to the Basuto tribe. And these investigators said that the brain of this microcephalic grouped with the brain of LB1. In these photographs are from the original description of this skull, which was done in the early 1900s. And in the summary of the article, it said that this individual had an ape-like protrusion of the jaws. And it certainly appears that the jaws are quite protruded. In the original description of LB1, it was stated that the protrusion of the jaws of LB1 was less than that of an Australopithecus. Here are two microcephalic skulls that we've used in our studies. To our eyes, the facial bones and the calvarium of these two microcephalics look a lot like this microcephalic and not like LB1. Dean this morning talked about this study in which we created classification functions for 10 normal humans and 9 microcephalics. These are in blue. Actually, we had 4 different ratios that we used. We had a lot of different classification functions. They all successfully separated normal humans from microcephalics. A simple plot of cerebellar protrusion and relative frontal breadth. Remember, microcephalics have the pointed frontal lobes and their cerebellums stick out behind the brains. Demonstrates that normal humans group together and that microcephalics group together. LB1, which is this endocast, groups with normal humans, and the Basuto woman or Lesotho woman groups with the microcephalics. This study was subsequently criticized because we had included juveniles, 4 juvenile microcephalics. Turns out that the brains of microcephalics have completed growth by about the age of 4. The small letter M stand for microcephalics. A grouping of the juveniles, to our eyes, is no different from the adults. This other individual, the brain is a little bit larger. In the museum collection, it was listed as a 3-foot-tall dwarf. It classified with the microcephalics. In our article, we said that we thought that it probably was an example of that condition I talked about earlier, MOPD2. Last year, a study was done. Actually, the focus of the study was the identification of the genes responsible for this condition. And in the last paragraph of the article, the authors suggested that actually they talked about Homofloresiensis in the last paragraph. In the next to last sentence, they suggested that Homofloresiensis may have had this pathology. Here are a couple of images from their article, and compared with LB1, people with MOPD to have, they have less sloping of their forehead compared with LB1. They appear to have chins. LB1 has no chin. They don't have supraorbital tori. LB1 has supraorbital tori. And their teeth are small and widely spaced, unlike the teeth of LB1. Curiously, 2 years earlier, a very detailed study, statistical study, had been published, and in that study, MOPD2 was considered, and a whole list of traits were given that separated MOPD2 from LB1. Two of these were that LB1 tends to have long forearms. People with this condition have relatively short forearms, and as Bill just mentioned, the hips of LB1 are wide and flaring. People with this condition have narrow hips. Also, these authors referred to this article in which 58 subjects had been studied, and in that article there are a number of traits that would separate MOPD2 patients or people from LB1, but we think the most important is that there's never been a documented pregnancy for a person with this condition. Another pathology that has been suggested for LB1 is the Larson syndrome. In their article, the investigators used 33 diagnostic criteria for which they said there was a similarity between LB1 and people with this with Larson syndrome. 32 of these traits were given in the original descriptions for Homofloresiensis. And I'm not going to go over all of them, but here are a few. Larson syndrome people do not have brow ridges. LB1 does have brow ridges. LB1 has a sloping forehead. Laron's people with Larson syndrome have bulbous foreheads, frontal bossing. The widest diameter of the skulls of people with Larson syndrome is high on the skull. That's caused by a narrow cranial base. It gives the skull's quasi-triangular shape. LB1's widest width is near the base of the skull. They provided this image, a lateral head plate, and said that Larson syndrome, people with Larson syndrome don't have chins. Well, maybe it was a little overexposed when the radiograph was made. Maybe it was overprocessed, or maybe the digitizing wasn't done properly. At any rate, we enhanced this in Photoshop, and there is a chin present. This is typical of lateral cephalometric radiographs on chins that are developing. You can see there are baby teeth, deciduous teeth in this individual. You can go to Image Google, enter lateral cephalogram or just cephalogram. You'll see many instances of chins that are developing that look like this. This chin is, to our eyes, is considerably different from the chin of LB1. As Peter Brown discussed today, these tori, the bossing, the massiveness of this jaw is, to our eyes, completely different from patients who have Larson syndrome. They also suggested that the head circumference of Larson syndrome patients is similar to that of LB1. This is a plot of age against head circumference. For normal humans, all of the markers are for patients with Larson syndrome. And in general, they do have smaller head circumferences than normal humans, but there is overlap. Here is LB1 by itself. And Peter Brown has mentioned these very unusual premolar teeth. The mandibular premolar teeth, those are the premolars, are the teeth right in front of our big chewing teeth or molars. These are massive, they're two-rooted, they're very unusual. The authors provided this image and said, here is a patient with Larson syndrome, and this patient has a two-rooted premolar. Here's a deciduous tooth. We can tell that's a developing premolar. Here's a radiograph that was taken at a dental school near where I work, and it has 2 roots also. When your teeth were developing, when my teeth were developing, when everyone's teeth around the world are developing, if you radiograph those premolars, they will appear to have 2 roots. They don't have 2 roots, they're conical in shape, but the lateral walls absorb more radiation than does the middle part of the tooth, so the teeth appear to have 2 roots. They don't. They are conical in shape. And again, if you go to Google and enter dental radiograph, you'll see dozens of radiographs such as this. The next pathology that was suggested for LB1 was Cretinism, and the authors suggested that LB1 had a number of features in common with cretins, including open anterior fontanelle, which was discussed earlier today, missing permanent teeth, and retained baby teeth or deciduous teeth, and enlarged pituitary fossa. Well, as soon as I saw this article, I went to Kirk Smith and I said, gee, wasn't that area on the skull, isn't that the damaged area? And he said, yes, I think there is a damaged area, anterior fontanelle, open anterior fontanelle, and we looked at the CT scan. It certainly appeared to be damaged to us. I think this edge is 8 or 9 millimeters thick. I emailed Bill Jungers that day, and he said he had just gotten an email from Peter Brown, and as we heard earlier today, the missing part of the skull is back in Indonesia. The authors referred to this article in which these 2 images appeared. This is a retained baby tooth or deciduous tooth on the left side of a person's mouth, and here's retained baby tooth on the right side of the person's mouth. And they suggested that these retained baby teeth are the same as LB1's premolars. There's no similarity. These look like deciduous teeth. The root structure looks like they should. And they're small. They have short roots. They do not look anything like LB1's premolars. We concluded that LB1 did not have. Then their final piece of evidence was that they made measurements of the pituitary fossa of LB1, and they made these measurements from 2-dimensional images that we had published in our original article, and they got a measurement of 12.9 millimeters. We looked at the CT scan data. Here's the pituitary fossa. Here's a 12.9 millimeter measurement. It does not fit. The longest measurement we could make for the pituitary fossa was 9.76 millimeters, which is in the range of normal humans. We concluded that LB1 did not suffer from cretinism. At times, this has become a bit strange. Last year, this book was published, and in the book, it was suggested that LB1 had a filling in its mandibular left first molar, and that this filling had been placed by a traveling dentist, and the filling was of unknown composition. Now, all of this wasn't in the book. The composition, I don't think it was mentioned in the book, but subsequently the author told me it was of unknown composition. When I originally saw the picture of this tooth, all I saw was the mandibular left first molar, and I couldn't tell much from that. As soon as I had a chance, I looked at the original photographs that were published with LB1, and what is obvious is that there's extensive tooth wear. The enamel is missing from the mandibular left first molar, but and the dentin is exposed. The same as dentin is exposed on the second molar and on the premolars, on both sides of the teeth and all of the molars on the right side, and all of the teeth in the maxillary arch. There is more exposed dentin or more severe tooth wear of this mandibular first molar on the left side, but this to me looks like exposed dentin. We studied the CT images, and dentists, when they place a filling, are trained to prepare teeth in a specific way so that the fillings will be retained. There was no evidence that a filling had been placed in the lower first left molar, and there was no evidence of any kind of tooth preparation or temporary filling. This whole thing didn't make a lot of sense to us because temporary fillings last typically, they're called temporary because they're temporary. They don't last for more than about a year. So if a temporary filling had been placed in this tooth, LB1 would have had to have died within a year, would have had to have been taken to Liangbua Cave and buried 19 feet deep. The CT scans indicated that the dentin is demineralized. Now, the bones, when they were covered of LB1, had the consistency of wet cardboard or blotting paper. Dentin and bone have about the same consistency, so it may be that the same processes that affected the bone affected the dentin, but we found no evidence that LB1 had a dental appointment. Now, with regard to all of the pathologies that have been suggested for Homofloresiensis, that's about par for the course. When Neanderthals were first announced as a new species, Homo erectus and Australopithecus africanus, it was suggested that their remains were nothing more than those of pathological or normal or humans with some pathology, or that they represented some aberrant or unusual ape. We found no indication for pathology for Homofloresiensis. Our conclusion is the skeletal remains of Homofloresiensis are not consistent with the pathological condition that persisted for hundreds of generations and thousands of years. We would like to thank Mike Morwood, Peter Brown, and our Indonesian colleagues for access to the CT scan data, National Geographic, and Mallinckrodt Institute of Radiology.
Thank you, Charles. It's amazing. Now I understand the length of your publication record. Responding to the Disease of the Month Club would keep you very busy. I can also attest, having seen a few deciduous teeth in my time, those are not deciduous teeth. That concludes today's symposium. The speakers, the 9 speakers from today, will assemble a little bit later on the stage for an open panel discussion, open questions, and discussion of issues. It's now my pleasure to introduce the president of Stony Brook University, Dr. Shirley Strum Kenney. Dr. Kenney has served as president for 15 years. She's, like Bill, also a native of Texas. She's transformed this campus into a living and working environment that's a pleasure to inhabit, and has brought Stony Brook recognition as a national leader in public higher education. In particular, she has presided over the development of the Turkana Basin Institute, and through its auspices has brought us the Stony Brook Human Evolution Symposia. This is the 7th such symposium, which means that Dr. Kenney has played host to more paleoanthropologists than she probably cares to remember. Madam President.
Thank you very much. I just want very briefly to welcome all of you here for this 7th Human Evolution Symposium. It is a very special privilege for Stony Brook to host these annual events. And indeed, it is a very special privilege for us to have the Turkana Basin Institute, the Symposium, and all the exciting things that are happening. If you are a groupie, as indeed I am, this is absolutely delicious. And the fact that we have with us such incredible world-class scholars in the field working together, giving us the various facets on each year's topic, and certainly the very fascinating Hobbits, it's something very special, and we're glad you could participate with us. We are glad that we are able to host these world-class scholars, and we are particularly glad also that on our faculty is the person who started all this. We have always had a great physical anthropology department and remarkable scholars in paleoanthropology. But Turkana Basin started with our very own Richard Leakey, and I am delighted to welcome him to come to the podium and start the next part of our meeting, and also Meave Leakey, who is another member of our faculty. We are very proud of these newest members of our anthropology department. So, Richard?
Well, thank you all very much. Thank you, Shirley. We will have an open panel discussion where I think a lot will be said, but I'm asked to present some of the issues and perhaps put some of the things that have been presented today into a slightly different context. The first thing I'd like to say is that I think we should all be very grateful that we're not here as a result of jury duty, because I think if we were, we were going to be here for some weeks. I think the evidence that has been presented is fascinating. I think there will be people in the audience who have many questions, but whether we could reach a conclusion that would be satisfactory to the majority of us is I think, unlikely. The second thing I would like to point out is that, and I think needs further discussion, is that at the moment much of the presentation was, to my thinking, lacking in a certain depth and breadth. For example, I think there are a number of people who recognize that AL288, the Lucy skeleton, is not necessarily representative of the genus Australopithecus. It's been called Australopithecus. But there are now, and I'm sure some people in the room know, that there are one, possibly two, almost complete skeletons that will be brought to light in the next few years. And having seen one of the two, I think we're going to be surprised that another Australopithecus isn't necessarily the model that we are using in this discussion. Secondly, I think there is a question mark in my mind in that we have referred to Homo erectus, and I think one of the question marks that we have to keep in mind is that we don't actually have any Homo erectus feet. And the suggestion that Homo erectus left footprints 1.5 million years ago in East Africa is but a suggestion. It was at a time when there were three different hominid taxa, and based on the occurrence or the commonness of fossils, the Homo erectus is one of the rarest of the three there, and in probabilities it wouldn't be Homo erectus but something else. So I think there are a number of much deeper discussions and issues that we should consider in this fascinating new hominid and hominids from Flores. I myself have not spent a great deal of time reading the literature, and I was particularly looking forward to today's presentations. And I conclude this workshop or seminar with a far greater degree of persuasion than I came in with this morning, and I think that speaks for itself. But I think one also needs to consider that there are some very puzzling questions, whichever way you come at this, and I don't think that we have sufficient evidence to answer some of the questions that I think are most taxing to some of us in the audience. I would like a little more discussion about the dating. There are some tophaceous layers. I think in the reference to the dating that was made at the beginning of this meeting, reference was not made to radiometric dating of anything taken from the tophaceous material. If it was, I didn't hear it, and I'd like a little more discussion in the panel concerning that issue. Secondly, we've referred to the skeleton as it should be, and the number 6 in this discussion, but I think there are obviously other elements and other specimens which I would like to be brought out into the discussion to a certain extent, because I think there's some lingering question, well, what about the others? And I think that's important. I think the third thing that I would want to see more discussion about is the linking of the lithic material, the stone artifacts, to this discussion. And whilst that I think it is important that there are stone artifacts there, I think we also have to remember that anatomically modern humans appear long before this, and I think that was alluded to back almost to 200,000 years ago in Africa. But it was also mentioned that Homo sapiens, modern Homo sapiens, got to Australia 50,000 years ago, and I think there is an issue there that needs further discussion. And in particular, I'm interested in the geographic isolation of Flores, in terms of when people could have got there from land bridges, or if there's no possibility of land bridges, what the possibility is that what the basis is of supposing that they came in one time or another, and what is the geological information on that. And I would hope that could come out of further discussion as well. I think the possibility of it being a legitimate new species has been greatly strengthened, and I thought some of the discussion, particularly about the shoulder and the wrist, was fascinating stuff. And I would like to have possibly a little more discussion about the implications of the absolute length of that foot. And I liked your moving video of this shuffling hobbit. But I have thought of the hobbit operating like we do when we walk to the beach in flippers, and not a particularly satisfactory way of getting, and I could imagine flippers on your model. But that has to have had implications to long-term survival, I would have thought, although obviously if you take carnivores out of the formula, as you mentioned, there's less need for speed and mobility. I don't think I can add more. I would love to ask some questions from the audience as part of the audience, not as the convener. And before I remove myself from the stage, let me just pay a personal tribute to all the speakers who I think have presented a very solid piece of work. And I only can think in terms of my own experiences in paleoanthropology, and dramas come and dramas always go. And don't worry too much about the cheap shots, some of which will prove cheaper than others, and some of which will pass into oblivion. And if you're right, you can remind people. And if you're wrong, people generally forget faster than you realize. So take good cheer from a man who's had some experience at presenting controversial material, and a family that has probably accumulated more experience in controversy and paleoanthropology than possibly any other family. So I thought that the last contribution was a little bit defensive. A pox to those who aren't here. Don't worry, you presented your side of the story, and we're interested to ask you some questions now. Thank you all very much, and may the panel now come forward.
Thank you, Richard. If we could get the 9 speakers from today to come up and take a place at the table. Any order you'd like. We have 2 volunteers with microphones. If there are questions from the audience, we'll do our best to get the microphone to you. If it's not possible, I will attempt to repeat the question if I can remember it. Or if I can't, I'll ask my own version. One of the things I found most fascinating from today's presentations was the notion that most of what we teach in human evolution or have thought about in human evolution with regards to Homo erectus originating in Africa and emigrating out of Africa at about 1.5 million years, striding out on its long legs, may have to very well be rewritten. Not only does the evidence that was presented here suggest that something more primitive than Homo erectus, something resembling Homo habilis, may have left Africa earlier, but there's also evidence from the Georgian site of Dmanisi that was mentioned, where we have Homo erectus-like forms that are about 1.8 million years, and work by colleagues at Binghamton University, Philip Reitmeyer, have quite convincingly demonstrated that these Homo erectus specimens actually retain a number of primitive features that are exhibited by crania that we classify in Africa as Homo habilis. Philip and I believe some of the panelists here would have suggested that maybe something like Homo habilis actually left Africa, and that Homo erectus may have evolved in Asia, exemplified perhaps by Dmanisi, and then moved back into Africa from Asia, so that we may have a out of Africa, back into Africa scenario with the populations, am I lecturing? With the populations of this early primitive form then subsequently evolving in isolation, such as in Flores, to represent these species Homofloresiensis. Would there be anybody on the panel that would like to comment on that? That was a long speech, not a question. That was my statement of fact.
Which part of it, Fred?
about the notion of something more primitive.
I would say Dmanisi already exhibits the modern body plan in terms of elongated hind limbs, and what's known of the feet suggests that it has it's modern in aspect. What's unusual about Dmanisi is, compared to the so-called strapping youth, is its body size. I think that's been the surprise. And also the fact that at least one individual has a 600 cc cranial capacity. There's certainly features that are primitive about that skeleton, but in terms of the locomotor skeleton, I think we're already looking at something that is remarkably different than Homo floresiensis. And there are going to be a lot of vertebrates that are going to be surprised that you need to evolve long legs in order to get out of Africa.
I think Philip Reitmeyer was talking about the cranial morphology at Dmanisi showing a whole suite of primitive features, so that we may have something that's actually got a mosaic.
Can you raise the volume slightly?
So what I think he was referring to, or I'd be alluding to is a mosaic at some place like Dmanisi, where you've still got primitive retained features, but something that's evolved more erectus-like condition, which would suggest that somewhere in Asia we have a Homo habilis-like form that just remains to be found, which you might say, well, there's no evidence for it, but then we don't have a lot of evidence, period.
We didn't have evidence for Dmanisi until it was discovered either.
It's true.
But as it comes to skulls and teeth, I would defer to Peter.
Thanks for dropping me in that. Dmanisi shows lots of variation. Some of it appears to be sex-based, some of it has to do with ontogeny and growth of Dmanisi. And I think the interesting aspect of Dmanisi to me, it's sort of convincing me that the Flores people, or the ancestors of the Flores people, probably on balance came out of Africa before the ancestors of the Dmanisi people. Mainly because, as Bill said, the Dmanisi body proportions and the way their bodies worked appear to be more modern in terms of function than those which we finding on Flores. And yes, there are some things in common between the Flores people and those of Dmanisi, like in premolar morphology and root morphology and the shape of chins, but there are other individuals of Dmanisi who don't have those characteristics as well. It just depends on which ones you want to push.
Can you not hear the speakers? Okay. Are there any questions from the audience? Oh, Paul? Wait, can we get a microphone to Paul?
Well, I have a loud voice too. There's a question I wanted to ask Mark earlier after his talk, and I've given him advance warning of this, so he has time to think about it. I thought it was a superb study of the lithics. The study itself, the way you presented it, was a brilliant piece of analysis and presentation. I was just left with the one question, as you know. Granted this extraordinary similarity and apparent continuity in technology before and after about 11,000, how does one interpret that? You were suggesting that it was the modern humans coming in after 11,000 who were sort of being heavily acculturated, if you like. You want to use that term, or picking up the local technology. But the big question which has to be asked, how do we know that after 11,000 there were modern humans who were making those tools? I mean, if the Hobbits could survive for 800,000 years, couldn't they survive for 817,000 years?
Actually, that's a very clever way to phrase a question that I've received from a lot of people, actually. Now, I think it's important that I emphasize right from the start that my premise was that the hominid bones that were found in association with the tools were the hominids that made those tools. And I think when you start backing away from that premise, that's the most parsimonious way to associate tools with hominids. And when you start backing away from that, I don't think that's necessarily a good approach at this stage of our analysis. We have one site, and this is the only place that we have Homo floresiensis.
I wasn't suggesting that, Mark.
Yeah, but what I'm saying, though, is that we have modern humans associated with those tools after 11,000 years ago. And we have Homo floresiensis associated with the tools before 11,000 years ago.
We do have Homo sapiens, modern humans, associated with that same technology immediately after 11,000 years ago, did we? We have skeletal remains.
Yes, actually, that's one thing that I
didn't know that.
I might defer to some of the people further down the table about the exact distribution of the Homo sapiens remains in the cave. A lot of the burials are actually much higher, the articulated burials, but there are individual Homo sapiens bones further down the sequence. But another thing that I'd like to point out is that at Liang Bua we have I mentioned it in the paper, we've got 80,000 years of technology in the cave, in the Pleistocene, with very little of the sorts of changes that are typical of modern human behavior elsewhere in the world. I tried to point out, after 11,000 years ago, the signature becomes almost immediately after 11,000 years, becomes very similar to behaviorally modern human sites elsewhere in the world, with the differences, preferences in raw material types. Those edge gloss flakes, if those other researchers are correct that those are from making baskets and mats, that's a very modern human sort of technology that we don't have any evidence for with earlier hominin species. And there's other materials in the cave that don't involve lithics that suggest modern human type behaviors, with marine shell being imported from the coast. I mentioned metallurgy, all kinds of very modern human looking behaviors there. The other issue that's interesting about Liangbua as well, the question that I kind of expected, which is why I thought yours was a clever way of phrasing a similar sort of thing, is, well, how do we know that Homo floresiensis made tools full stop? Maybe modern humans made all the tools in the cave. The problem with that is that continuity that goes all the way back to Mare Menge, which is before modern humans even evolved, let alone to just the early occupation of the cave at 100,000 years ago. That's nearly double what most models call for, or many models call for the spread of modern humans out of Africa. Does that?
Yes, I didn't realize you had anatomically modern humans immediately above that 11,000 year interface. I didn't know you got the direct association of that primitive continuing technology with anatomically modern humans. Of course, the other possibility is that if it was the same, if it were the Hobbits continuing with 90% of their primitive technology, the bits of modern human technology you see with it, of course, it could be, as with the Neanderthals, possible acculturation from modern humans as opposed to modern humans doing it. That's another hypothesis, but thank you for those observations.
Any other questions? John? It's not on. No.
Now?
No.
All right. This is another archaeology type question. If you're looking to diagnose presence of Homo sapiens in an island environment, one really good predictor is the extinction of all the other big mammals on the island. And what I want to ask you is that if in the sequence at Liangbua or any of the other caves, do you see that kind of punctuation event that might mark the arrival of our species on the island? I'm just thinking maybe it's a signature that is not lithic.
His question was, with the arrival of Homo sapiens on the island, do you see the extinction of large-bodied mammals?
Or does it precede? Do such extinctions precede the point at which you have Homo sapiens fossils in the cave? Does it go? Do you see an extinction at 45,000 or 70,000 or 80,000 that you might attribute to human activity?
I might defer to Mike's greater expertise on that.
Well, Stegodon becomes extinct at the same time as Homo floresiensis on the island. We got Stegodon down to about 17,000 at least. And then later on in the Holocene, Komodo dragon becomes locally extinct in the Liangbua area. So the arrival of modern humans does coincide with the extinction of the only large endemic mammal on the island. Mm-hmm.
Wait for the microphone, please.
Oh, you didn't point to me.
Oh, wait, one second, Jack. I didn't see you.
Okay.
I was wondering
the microphone's not on.
The shuffling of floresiensis. Could that be that perhaps the environment was sandy or water-like, and that's why they had the big feet like duck do?
The long feet are an illusion. They are long only in respect to the hind limb. If you use any other body size surrogate, whether it's humerus length, femoral head diameter, estimated body mass, the feet are not long. So again, many aspects of the proportionality you're seeing that is peculiar to Homo floresiensis is driven by an extremely short hind limb, which we don't have many of in the fossil record. And again, that's why the Lucy model is particularly helpful. So it's not, I wouldn't call it a shuffling gait, I would call it a high stepping gait. And it turns out that long feet for short femora, it's not such a bad thing for walking. There are people in this audience who could tell you about the collision mechanics of bipedalism. It turns out that for walking gaits, big feet are pretty good. The problem is those long toes and long feet would be terrible. They're poorly designed for running, particularly endurance running. And so the signature of short toes that you see in modern people is a very derived feature. And quite honestly, we don't know when that occurs. I actually find the Homo erectus footprints to be a bit more compelling than Richard. And the feet of Dmanisi, if you're willing to grant them Homo erectus-like status, suggest the same thing. So again, it's not a shuffling gait. It's a perfectly adequate bipedal gait. It's just they're not marathoners or they're not sprinters. And I'm sure they did just fine wandering the landscape of Flores Island.
Jack?
Can you hear me? Yes. I have a question based on something Dean Falk said, but it deals with the postcrania, so Dean, you may not be the one who wants to answer it. You said that the innominate bone, the os coxae, looked like Lucy's. And other than iliac flare, were there any other traits about the os coxae that resembled Lucy's?
I defer to Bill Jungers.
The bony pelvis is we're lucky that we have the bits that we have now. It suffered a bit in its trip outside of Jakarta, but I think what Peter Brown and colleagues described initially is accurate. It's not only iliac flare, but it's also a fairly anteriorly placed iliac pillar and a relatively small acetabulum. So the overall package has some aspects that recall Australopithecus as we know it, whether it's STS-14 or Lucy. We haven't been able to assess how the orientation of the iliac blade in terms of the gluteal surface, because that area is very eroded. But that's obviously something that we have CT scans now for, and hopefully we can assess to see, do you have a Lucy-like condition or an STS-14-like condition, Africanus, where the gluteal surface tends to face more posteriorly. I don't know the answer to that, but there are other features other than flare that suggest it's not a modern pelvis. And by the way, it sure looks like a female pelvis based on the sciatic notch.
There's a fellow up behind Jack.
Hi. A number of the talks had implications for, very kind of contradicting implications for hunting culture in Floresiensis. On the one hand, Mark Moore mentioned the presence of perforation points, which might or might not be connected to weapons, but then Bill Jungers and Susan Larson both mentioned mobility limitations in running and throwing that would make hunting difficult. Is there, what kind of archaeological evidence is there in the living sites for Floresiensis hunting, like prey animals or anything like that?
I think the evidence is certainly for butchering, and just how the juvenile stegonon and Komodo dragons were actually obtained, we don't know, but certainly there are cut marks on Komodo dragon bones, on large bird bones, and on stegodon bones from the site. So we can definitely say butchering. Hunting, maybe scavenging, we're not certain at that point.
And if I can just comment on the perforators, they're few in number, and there's no impact damage. There's actually attributes we can look at on stone projectile points to see if they were used as projectiles. And these perforators have no signs of any of use of that nature. And frankly, a lot of the retouching techniques form a continuum with these items that we called perforators, it's possible that those are just retouched edges that happen to isolate an area in between that looks like a perforator, and that's why I'm really cautious about those tools. Of course, if they were some sort of projectile point, that would imply hafting, and that implies all sorts of cognitive issues that I think are unwarranted to go on just based on the present evidence that we have.
I just add some information. Now we just analysis about the stone artifact and residue, and still in progress. Some of them we found the bloods and also the fat and also the resin, so that soon we will have the information about that.
Sorry, Brian, I didn't realize it was you, the fellow sitting behind Jack. I can't see because of the lights. Any other questions? Yes, that's the... Yes.
Okay. Dr. Hildebolt, if that's the correct pronunciation, you mentioned that there is a population of pygmies on the island of Flores. Did I understand that correctly?
That's correct.
Yes. It seems that that's a very important point to me. It was sort of, brought in at the end of the day. I had never heard it before. But how extensively have they been studied could there be any evolutionary evidence there that they are descended from this?
Yeah, well, others here could probably answer that better than I could. I meant to mention, one thing that I found curious, some of us visited that village in 2007, and I happened to be standing behind several of the skeptics, and I overheard one whisper to the other and said, well, gee, I thought these pygmies were going to be shorter than they are. And I actually had a picture. Now, I'm short, but I'm not pygmy short. But some of the pygmies were taller than I am, and they were considerably taller than some of the skeptics. But Mike, maybe Peter or Bill could answer how extensively they've been studied. In the PNSA article by Jakob, they supposedly studied them, but I'm not quite sure we can say that was an extensive study.
As perhaps some of you know, there hasn't been a publication on the human biology of these individuals yet, but there was an abstract presented at the American Association of Physical Anthropology meetings this April, and it was supposedly about short people on Flores, and it was a sample size of 3. And it was, one of them was almost certainly diagnosed as an achondroplastic dwarf, and the other 2, they were unsure. They thought they could be related, and so they needed to do endocrine work. But the data that was reported in summary fashion by Jakob et al., again, there are some short people on Flores, but they are certainly nowhere near the size of small-bodied peoples elsewhere in the world. As pygmies go, Charles is right. They tend to be kind of big pygmies.
Thank you.
Richard?
Can you hear me? Yes. I just wanted to ask the panel if somebody could explain or clarify or get me to understand. Looking at the photographs of the cave floor today and where you're working, and the photographs of your many-meter deep pit where the hominids were found, I can't remember whether it was 19 meters or some figure in that area. What was the basis of locating that deep pit there, and have there been any other test excavations to see what the profile might be at that level in terms of cave fill and things of that kind? And what is the distribution through the 19 meters of vertebrate and lithic remains?
We've excavated to a maximum depth of 12 meters. The initial choice of areas to be dug where people had previously dug. So rather than having to dig down through modern and Neolithic and that, emptied out backfill and then continued on down. It's a way of, I guess, conserving the deposits. So the actual skeleton was taken out from a square that had been previously excavated to a depth of about 3 metres, and we continued that excavation to a depth of 12 metres and found the skeleton at 6 metres. Nowhere have we found bedrock in the site, and probably the deepest, I mean, it's quite a complex geomorphic history, and the deepest actual cultural deposits are about 12 metres in one section of the site. Does that answer your question? Okay.
Here. I didn't hear anybody speaking about the knee and the patella, especially the shape, the size, and the height of the patella relatively to the middle of the joint, the knee joint, and its influence on walking and running.
There are two patella. They're remarkably similar to modern humans in most respects. They have sort of the same height and width proportions. They even have the convenient notches on the lateral side that allows us to tell which is left and which is right. They bear no resemblance at all to the sort of discoid ape patella. So you may not be aware, there are not a heck of a lot of patellae in the hominid fossil record to compare these two, but they're certainly compatible with the knee joint, which has the typical inward sloping orientation that we see in modern humans. I don't want to give the impression that these were somehow clumsy bipeds. They have the full suite of bipedal features we observe, and part of that suite is, in fact, a human-like patella. The one aspect of their patella that is a little different than humans, it's not quite as beveled, not quite as angulated on the articular surface. But other than that, you would never confuse it for anything but a human-like patella.
This fellow there.
Yes, just a quick two-part question. I know we talked about the shoulder and the wrist, and it was mentioned that throwing may have been a very important adaptive behavior. So my one question is there any evidence that throwing was a major force behind the development of the arms that we have today? And the second part of my question has to do with the stone tools. I noticed, and I only saw what we had on the slides, there was a similarity through all of the different stone tools back for hundreds of thousands of years with a circular cut piece that looked like it was sharp on all sides. Didn't look like it would be ideal for butchering because your hand would be against the sharp edge. On the other hand, it would be ideal for anybody that skipped stones on a creek or a pond knows that it would be ideal to fling that way. And it's flung sidearm. You would not have to have an arm and structure developed to throw like a major league baseball player. Throwing sidearm is somewhat easier, and while it wouldn't have the accuracy, the spinning stone would also tend to stay the way it is gyroscopically because of its spinning, and might have been, has there been any speculation on that?
Well, I have to admit, I'm not as familiar with the forearm as I am with the shoulder, so I can't say whether there are throwing characteristics in the forearm. There are forearm elements known from Flores. The LB1 skeleton includes a almost complete ulna and from one side and a partial ulna from the other side. There is a ulna associated with the LB6 individual, and there is another ulnar fragment from much earlier. I believe that's LB3. Bad with numbers. But in any event, there is material. There are several radial pieces as well. So there is material for which people, if they're interested, could study the forearms of the Flores hominids. Now, in regard to your question about sidearm throwing, I mean, I did picture overhand throwing, but I want you to imagine the shoulder as if you had a cone which described the range of motion. And, of course, the apex of the cone would be the socket for the shoulder joint, the glenoid fossa. So if that cone is facing forward, I don't know the exact diameter of the cone, but I don't think it would include a motion that would be posterior to, say, the midline of the body. I mean, I don't believe that you can get a cone from that shoulder joint that would include a motion, a sort of extension motion behind the body. And if sidearm throwing entails doing that motion, I doubt Flores could perform that kind of throw either. Now, I also want to emphasize, however, that the range of motion that I imagine for the Flores hominins and for early Homo, as evidenced from the Dmanisi material and from the Narukatomi skeleton, would be completely compatible with making tools, with doing lots of different kinds of manipulatory behaviors, just not ones that entailed a sort of a posterior extension, especially of a arm that was elevated at the side. So I think that was the main limitation of this shoulder configuration. In regard to whether or not discoidal type tools could be used for cutting or butchering or other sort of things, I would have to defer to my archaeologist colleagues.
There actually is a literature, very obscure literature out there about hand axes, because it has been proposed in the past that those teardrop-shaped bifacial tools were made to throw. Now, things like that are very hard to test empirically. As far as the bifacial radial cores that we see on Flores, in Liangbua Cave, we sometimes actually find the flakes that were taken off in making these tools and can conjoin them back onto the cores. We haven't looked into that to a great deal, but that doesn't seem terribly likely if they're making these things to take outside the cave and throw them at things. Personally, I just can't really see the evidence that the cores were produced for throwing.
I'm going to take a stab at this as well with regard to the wrist, because I think when I talk about the wrist of Homo floresiensis and also the implications this has for how we look at hominin evolution in general, the questions always come up about behaviors, about stone tool making, throwing, and how could it do things if it doesn't have our hand, and these sorts of questions. And I think the fossil evidence is very clear as it stands right now, that we know that stone tool behaviors show up long before we see our hand. Now, that doesn't mean that we don't see changes to the hand during hominin evolution that are probably adaptations for types of manipulative behavior. However, we don't see sort of the derived structures in this part of the wrist until much later. So clearly there's a disjunct here. So we have to accept that the same is true in any vertebrate evolutionary trajectory, where animals are behaviorally flexible. And sometimes behaviors arise and originate long before any particular morphological specialization results. And it's mainly the fact that these behaviors are adaptive that it turns into contexts where now those variations in morphology have a chance to be selected on. Now, in the case of our wrist, it's very clear that we have not only Oldowan types of tools and all the behaviors that we may associate with how they're made or how they're using them, but also Acheulean hand axes. And through all of that, we don't see this. And so clearly, you don't need this to do that. But the fact that we're all here, and if we were to look at our wrist bones, we have the anatomy of a hominin that is descended from a nested groups of hominins, where first you get behavioral adaptation, which creates a context in which things like perhaps our shoulder and our wrist have been secondarily modified for. And why Homo floresiensis is so exciting in this instance is because it demonstrates to us that that is the case. That here you have a hominin that has the primitive morphology of the same types of morphology that we should expect to find in wrists that are between 1.5 and 2.5 million years ago. And so again, it shows that there was nothing wrong with the way their hand and shoulder and wrist and bipedality function. It just did it in a different way than we did. And the only reason we're here is because we've gone through secondary modifications to those anatomies relative to those behaviors. But the behaviors clearly evolved first.
Ladies and gentlemen, it's now 5:30. That brings us to a conclusion of today's symposium on the Hobbits. I'd like to thank you all for your interest and attention, and I'd like to thank the panelists for their perseverance and their wonderful talks. Thank you very much.